Assalamu alaikum, friends. I hope that you all will be fine. Friends, in this video, I'm going to discuss about an important topic in protein metabolism, and that is ammonia metabolism. Friends, it is a very important topic as a long answer question and as a short answer question. Okay, and I will cover this topic ammonia metabolism under the headings of sources, storage and transport, functions, disposal, and its toxicity. Okay, so let's start our lecture. इससे पहले अगर आप चैनल पर न्यू हैं तो काइंडली आप इस मेरे चैनल को लाइक भी करें, शेयर करें और सब्सक्राइब करें. So let's start our lecture with the sources of ammonia. Sources. The first one source of the ammonia is transamination and deamination of amino acid. This is the first source. Transamination and deamination of amino acid. Now, what does it mean, the transamination and deamination of amino acid? So, let's discuss about the transamination. That is transamination. This transamination means the transfer of amino group from amino acid to keto acid to form new amino acid and new keto acid by SGOT and SGPT. Okay, transamination means transfer of amino group from amino acid to keto acid to form new amino acid and new keto acid by SGOT and SGPT. So this is the definition of the transamination, and this transamination process, it takes place in cytosol. Must remember, this transamination process, it takes place in cytosol. And friends, this SGOT, that is serum glutamate oxaloacetate transaminase enzyme, and another name of this serum glutamate oxaloacetate transaminase is aspartate transaminase. While this SGPT, serum glutamate pyruvate transaminase, the another name is alanine transaminase. तो यहाँ पर गौर कीजिएगा आप, दिस इज द aspartate and alpha ketoglutarate. This is the amino acid and this is the keto acid. With the help of this SGOT enzyme, and the name is aspartate transaminase enzyme, and this enzyme requires PLP, that is pyridoxal phosphate, and it leads to the formation of new amino acid and new keto acid, and that are glutamate and oxaloacetic acid. Okay, so there is a transfer of amino group from this amino acid to this keto acid, and there is a formation of this new amino acid and new keto acid by this enzyme, and this enzyme requires PLP, that is pyridoxal phosphate. Let's see another example, that is alanine and alpha ketoglutarate with the help of SGPT enzyme or alanine transaminase enzyme. This reaction also requires PLP, that is pyridoxal phosphate, and there is formation of glutamate and pyruvate. Okay, so this is the transamination process. There is a transfer of amino group from amino acid to keto acid to form new amino acid and new keto acid by this enzyme. So this is the transamination. Now coming toward the this deamination of amino acid, that is deamination. We can say that oxidative deamination. The definition is the coupling of oxidation with deamination. It is called as oxidative deamination. The coupling of oxidation with the deamination. Deamination means removal of deamination mean removal of ammonia. This is called as deamination. Okay, now let's see with the example. This is the glutamate. This glutamate, it converts into alpha ketoglutarate, and there is removal of ammonia with the help of glutamate dehydrogenase enzyme. Glutamate dehydrogenase enzyme, and here NAD plus, it converts into NADH and H plus. So this is the oxidative deamination process. The coupling of oxidation with the deamination, it is called as oxidative deamination. This is the oxidative deamination, and this process takes place in mitochondria. So both are very important as a short question and as well as for point of MCQs. So this transamination and deamination of amino acid, it is the first source of the ammonia. Now coming toward the another sources of the ammonia. Okay, take a screenshot. Okay. Now the another source for the ammonia, that is purine and pyrimidine. Okay, purine. The adenine and guanine, they are called as purine, while thymine, cytosine, and uracil, they are called as pyrimidine. They are very important in the synthesis of nucleotide. Okay, and they are also the sources of ammonia. Now coming toward the third source of the ammonia, and that is biogenic amines. Okay, now coming toward the fourth source, that is intestinal bacteria. This intestinal bacteria, this intestinal bacteria, it converts urea into ammonia, and this ammonia, it gets detoxified into urea by the process of urea cycle, and urea cycle process takes place in the liver, and we'll discuss the urea cycle in future lecture. So these are the sources of the ammonia: transamination and deamination of amino acid, purine and pyrimidine, biogenic amines, and intestinal bacteria. Now coming toward the storage and the transport of ammonia. Storage and transport of ammonia. Storage and transport of ammonia. Okay, friends, glutamate, this glutamate, it accepts ammonia. This glutamate, it accepts ammonia and it converts into glutamine by action of glutamine synthase enzyme. Glutamine synthase enzyme. With the help of this enzyme, this glutamate, it gets condensed with ammonia and then form glutamine. And this glutamine, it stores only in the brain and intestine. This ammonia in the form of glutamine, it stores only in the brain and intestine. Okay, so this is the storage point of view. While this ammonia as a glutamine, it is transported into the liver. I'm repeating, this ammonia in the form of glutamine, it is transported into the liver from the brain and intestine. Okay, so this is the storage and the transport of ammonia. Now coming toward the functions of ammonia. Okay, functions. Functions of ammonia. This ammonia, it is very important in the synthesis of purine and pyrimidine. Okay, it is very important in the synthesis of purine and pyrimidine. Okay, another function is, it is very important in the synthesis of amino sugar. Amino sugar like glutamate, N-acetyl glucosamine, N-acetyl muramic acid. These are amino sugars. So in the synthesis of amino sugar, this ammonia is very important. In acid-base balance, it is very important. In acid-base balance, this ammonia is very important. And in the synthesis of non-essential amino acid, the amino acid that synthesize in our body, they are called as non-essential amino acid, while amino acid that does not synthesize in human body but they are needed to supply to our diet, they are called essential amino acid. While non-essential amino acids, they are the amino acid that synthesize in human body. So these are the functions of the ammonia. It is very important in purine and pyrimidine synthesis, in amino sugar synthesis, in acid-base balance mechanism, and in the synthesis of non-essential amino acid. Okay. Now coming toward the disposal of ammonia. Disposal. This ammonia, it converts into the urea by action of urea cycle, and this urea cycle process takes place in the liver, and this urea, it gets excreted into the urine through kidney. So this is the disposal of the ammonia. This ammonia, it converts into urea through urea cycle, and that process takes place in the liver. And urea, it gets excreted into the urine through kidney. Okay, so this is the disposal of ammonia. Now coming toward the toxicity of ammonia. Toxicity. So this toxicity process, friends, toxicity, toxicity. Okay. Last point, toxicity. The increase in concentration of ammonia, it leads to the neurological manifestation. Okay, I'm repeating that increase in concentration of ammonia, it leads to neurological manifestation. Okay, neurological manifestation, the symptoms are slurring of speech, blurring of vision. So these are the symptoms of neurological manifestation. This increase in concentration of ammonia is very harmful. This increase in concentration of ammonia, it leads to decrease in concentration of the alpha ketoglutarate, that is intermediate of TCA cycle, and also leads to the decrease in production of ATP. This alpha ketoglutarate, it gets combined with ammonia and forms glutamate. So ultimately it leads to the decrease in the concentration of alpha ketoglutarate and leads to decrease in the production of ATP. Okay, this glutamate, it gets combined with ammonia and forms glutamine, and obviously the increase in concentration of ammonia, it leads to the decrease in the concentration of glutamate and ultimately leads to the decrease in the production of GABA. And GABA, it is the inhibitory neurotransmitter, gamma amino butyric acid, because this inhibitory neurotransmitter, it forms from glutamate and it leads to decrease in the production of GABA. Okay, and also this increase in concentration of ammonia, it also disturbs tryptophan metabolism, which leads to increase in the production of serotonin. We will discuss later in future lectures. So the increasing concentration of ammonia, it is very harmful. It leads to neurological manifestation. The symptoms include slurring of speech and blurring of vision. It also leads to decrease in the concentration of alpha ketoglutarate and leads to decrease in production of ATP, and also it leads to decrease in concentration of glutamate and leads to decrease in production of GABA, gamma aminobutyric acid, that is inhibitory neurotransmitter. So friends, it is all about ammonia metabolism, their sources, storage and transport, functions, disposal, and toxicity. It is very important as a long answer question and as a short answer question. If you have any query, you may ask in comment section. And kindly, it is a humble request to you that like, share, and subscribe to my channel. Keep watching and thank you.